Good morning, class. Today we're here to talk about two important processes of cell division, mitosis and meiosis. These will be easy to remember. Mitosis sounds like my-to-sis, and meiosis sounds like meiosis. Got it? Now, both mitosis and meiosis are processes of cell division in eukaryotic cells. Both processes begin with one diploid cell. Diploid just means that the cell has two complete sets of chromosomes to start with, but we didn't cut out all those chromosomes, just a few, so you could see the process up close. Why do we need two types of cell division anyway? Well, mitosis is for the growth and repair of somatic cells. These end up as cells in the body that make up things like the hair, skin, and nails. The process of meiosis is a little bit trickier and really important because it makes gametes. In females, these cells become egg cells through oogenesis, and in males, they become sperm cells through spermatogenesis. Both of these cells are necessary for sexual reproduction, to make babies. So just remember, meiosis is for gametes or egg and sperm cells, and mitosis is for the growth and repair of somatic cells. Both mitosis and meiosis follow mostly the same stages of division, but before they start dividing, they hang out in the stage known as interphase. Interphase is where a cell spends 90% of its time. This is when DNA is being duplicated, and kind of like Chompy here, the cell is collecting nutrients in order to grow and divide. This all happens during several stages called gap 1, synthesis, gap 2, and gap 0. Gap 1, or G1, this is where cell growth begins, followed by synthesis. This is the stage of interphase where DNA is duplicated. Gap 2, or G2, is next. This is where the cell doubles in size because the cytoplasmic contents double in size, shown here by our lovely models. Finally is gap 0, or G0. This is the stage after cell division when a cell can no longer divide. That's it for interphase. Now we can start with the stages of cell division. These stages are called prophase, metaphase, anaphase, and telophase. After mitosis completes one cycle of prophase, metaphase, anaphase, and telophase, it's finished. But meiosis is more complicated. It goes through those steps again, in meiosis too, but we'll talk more about that later. The first stage of cell development is prophase. This is where chromosomes condense, thicken, and in the beginning they coil up. The nuclear membrane also breaks down in the cell. In meiosis, something else really cool happens. Now don't freak out about this next big word. I'll explain. Homologous pairs of chromosomes meet up in the cell. Homologous just means that these chromosomes are roughly the same size and shape as one another. As the chromosomes condense and coil, they exchange some genetic information. This is called crossover. Non-sister chromatids of homologous pairs break off a small section of genetic information and switch it. That's all. And that's important to allow for genetic variations in each species. It's why we're all different. Here's another example of what that would look like if it was socks. On a more serious note, remember, crossover only happens in meiosis, not mitosis. When we get to the end of prophase and mitosis, we have chromosomes which are thickened, condensed, and have uncoiled. In meiosis, the chromosomes have also exchanged genetic information. That's pretty amazing. At the same time all of these processes are happening, another specialized part of the cell has begun to form. It's called the spindle apparatus. Now, in plant cells, the spindle apparatus isn't really visible under a microscope, but in animal cells, it is visible as a really cool starburst shape. no, not like that. More like this. Okay, now let's talk about metaphase. In metaphase, chromosomes line up at the equator or the middle of the cell, preparing to separate. This also happens to be the center of the spindle apparatus. In mitosis, chromosomes line up single file along the equator. In meiosis, homologous pairs are lined up together. See, aren't they cute? That's it for metaphase. This next stage is pretty exciting. Anaphase is where the spindle apparatus comes in and rips the chromosomes apart in a cellular tug of war. Or maybe it looks more like a dance, like this. During anaphase and mitosis, chromosomes are pulled apart, turning them into single chromatids. Then they begin to travel to opposite sides of the cell. These are called poles. In meiosis, homologous chromosomes are separated into single chromosomes again, and they too travel to opposite poles of the cell. The last phase of cell division is telophase. This is when chromatids or chromosomes have finally reached opposite poles of the cell. A new nuclear envelope has formed, and the final cytoplasmic separation, known as cytokinesis, completes. Here's a fun fact. In plants, cytokinesis completes at the cell plate, modeled here by Sharon Rose. The cell plate is formed because of the plant's cell wall material, and when it breaks down. In animals, it completes at what is called the cleavage furrow, because that is where the cell is cleaved in half. At the end of telophase and mitosis, we are left with two cells that are genetically identical diploid cells, and the cells at the end of meiosis 1 are genetically varied diploid cells. This is where mitosis ends. The process of meiosis 2 starts with interphase, which you remember really isn't a part of the cell division process of meiosis, but the stage before it begins. Cells thicken up and gather nutrients for a second division. In prophase 2, chromosomes condense and thicken. The nuclear envelope breaks down again, and the spindle apparatus begins to reform. It's really important, though, to remember that there is no crossover in prophase 2. Just uncoiled homologs at the end of prophase 2. In metaphase 2, the genetically varied chromosomes line up at the equator of the spindle again, just like in mitosis. But don't get confused, this is meiosis 2. The next stage after metaphase 2 is anaphase 2. This time, chromosomes are pulled apart by the spindle apparatus and become individual chromatids as they travel once again to opposite poles of the cell. Last but not least is telophase 2. The two diploid cells we started with have become 4 haploid cells. Haploid means that the cell has half of the chromosomes of the parent cell. Each cell is now genetically varied from the next. Once again, this is important for variation in the species and completes meiosis 2. Hopefully this gave you a little overview of the processes of cell division we have come to know as PMAT, or prophase, metaphase, anaphase, and telophase.
She's alive! Alive! Science.
Oh.
from my hand. Why don't people understand my intent? From my hand, why don't people understand my intent?